Good morning, class. Today we'll start with a new chapter that is metabolism. Metabolism means breakdown of substances or molecules or chemical biochemical reactions to produce energy. Metabolism has two words confined in it, that is anabolism as well as catabolism. Anabolism means synthesis of certain molecules while catabolism is breakdown of these molecules. When we when we start with metabolism, the first chapter we consider is carbohydrate metabolism. Carbohydrate metabolism, in this carbohydrate metabolism chapter, we'll be considering about the production, that is anabolism, as well as catabolism of glucose molecule or carbohydrates. The first pathway come across is glycolysis. Now, what is glycolysis? Glycolysis has two words confined in it, that is first glycus and second is lysis. Glycus means sweet while lysis means splitting. Now, glycolysis is a sequence of reaction that converts glucose to pyruvate in presence of oxygen or to lactate in absence of oxygen with production of energy in form of ATP. During this process, a molecule of glucose, that is a 6 carbon molecule, is degraded into 2 molecules of pyruvate, that is 3 carbon molecule. In standard condition, glycolysis is an hexagonic reaction that tends to be irreversible because of negative delta G. It is a unique pathway since it can function in both aerobic as well as in anaerobic condition. This pathway is also known as Embeden-Meyerhof pathway. It is a sequence of reaction which leads to conversion or breakdown of glucose or stored form of glucose, that is glycogen, to pyruvate or lactate with utilization as well as production of energy. The location of this glycolysis is cytosol. That means the enzymes responsible for this reaction are present in cytosolic part of cell. It is a major pathway in tissues lacking mitochondria, that is erythrocytes, corneas, etc. We already have studied the glucose breakdown into 6 molecules of CO2 plus 6 molecules of H2O with release of 2840 kilojoules of per mole energy. Now, the fate of glucose when it enters living system. Glucose is broken down into pyruvate by 10 successive reaction, that is glycolysis. Now this pyruvate can follow three paths, that is conversion into ethanol and carbohydrate, second is conversion into acetyl CoA, and third is conversion into lactate. When pyruvate is converted into acetyl CoA, it's an aerobic reaction, while when it is converted into lactate, it is an anaerobic reaction, and this reaction is known as Cory's cycle. Now we come to glycolysis 10-step enzyme catalyzed reaction. These 10 steps can be further broken down into two phases, that is first preparative phase in which energy is required, while the next stage is payoff phase, that is energy generating phase. In the first step, glucose is phosphorylated to glucose 6-phosphate by enzyme hexokinase or glucokinase, and ATP is required as a phosphate donor. Glucose and glucokinase and hexokinase are isoenzymes of each other. The basic difference between glucokinase and hexokinase is glucokinase is found in liver cells, that is hepatic cells, while hexokinase is found or present in extrahepatic cells. Hexokinase catalyzes this reaction and converts glucose into glucose 6-phosphate. In the next step, glucose 6-phosphate is converted into fructose 6-phosphate in presence of enzyme phosphohexoisomerase. It is a reversible reaction. In the third step, fructose 6-phosphate is converted into fructose 6-bisphosphate in presence of enzyme phosphofructokinase with utilization of an ATP molecule. The fourth step is conversion of fructose 6,1,6-bisphosphate to glyceraldehyde 3-phosphate and DHAP in presence of aldolase enzyme. The glyceraldehyde 3-phosphate as well as DHAP are interconvertible into each other by the action of enzyme phosphotriose isomerase. The fifth step is conversion of glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. The enzyme responsible here is glyceraldehyde 3-phosphate dehydrogenase. It also liberates a molecule of NADH. Since DHAP and glyceraldehyde 3-phosphate are interconvertible into each other, we will be multiplying the product as well as the energy equivalent released here with 2 during energetics calculation. This reaction is reversible reaction. Now the seventh reaction is conversion of 1,3 bis phosphoglycerate to 3-phosphoglycerate. This reaction is catalyzed by enzyme phosphoglycerate kinase. There is a release of ATP molecule as there is a removal of one phosphate from the system. In the next step, 3-phosphoglycerate is converted into 2-phosphoglycerate in presence of enzyme phosphoglycerate mutase. There is only change in position of phosphate from third carbon to the second carbon of the molecule. And it is also a reversible reaction. The ninth step is conversion of 2-phosphoglycerate to phosphoenol pyruvate. This reaction is catalyzed by enzyme enolase. The last and final step is conversion of phosphoenol pyruvate to pyruvate. And the enzyme responsible for this reaction is pyruvate kinase. There is a liberation of ATP molecule in the system. Now we come to the energetics as well as the product of glycolysis. We'll start from the first step from one molecule of glucose. It requires 2 ATP, it requires 2 NAD plus, requires 2 ADP plus 4 PI, which gives 2 pyruvate, 2 NADH plus 4 ATP plus 2 ADP and 2 PI. After balancing the above equation, we get one molecule of glucose plus 2 NAD plus 2 ADP plus 2 PI that gives 2 pyruvate plus 2 ATP and 2 NADH. 2 molecules of ATP generated can be directly used while doing work or synthesis. The 2 NADH molecules are oxidized in mitochondria under aerobic condition and the free energy released is enough for synthesis of 6 molecules of ATP by oxidative phosphorylation. That means 1 molecule of NADH gives 2.5 or rather 3 ATP molecules. Under aerobic condition, pyruvate is catabolized further into mitochondria that through pyruvate dehydrogenase and citric acid cycle, where carbon 1 atoms are oxidized to CO2. The energy released is used in synthesis of ADP, ATP, NADH, and FADH2. Now, there is a chart of energetics followed during glycolysis pathway. There are two sections. First is energy produced while and the second is energy consumed. There are 3 reactions where energy is produced in form of ATP and NADH. The conversion of glyceraldehyde 3-phosphate to 1,3-bis phosphoglycerate liberates one molecule of NADH. We multiplied this one molecule of NADH by 2, which means we get 6 molecule of ATP. The next step is conversion of 1,3-bis phosphoglycerate to 3-phosphoglycerate, which liberates 1 ATP, which is multiplied by 2 because of the interconversion of glyceraldehyde 3-phosphate to DHAP. The third step is phosphoenol pyruvate to enol pyruvate, that is, or we can say pyruvate, which releases 1 molecule of ATP multiplied by 2, that is 2 molecule of ATP. In this 10th step process, we required ATP twice in the system. That is first reaction, which is glucose 6-phosphate to glucose phosphate to glucose 6-phosphate, while the second is fructose 6-phosphate to fructose 1,6-bis phosphate. The total required and the total ATP count here is 10 produced and 2 is required. So 10 minus 2 is 8. Thank you.